Good morning. I am Dr. Yogesh, Professor and HOD, Department of Physiology, Sri Chamundeshwari Medical College Hospital and Research Institute, Channapatna. So next, this we are going to proceed with the WBC experiment. So in the WBC experiment, the name of the diluting fluid, what we use is the Turk's fluid. So Turk's fluid contains glacial acetic acid, gentian violet and distilled water. So what glacial acetic acid will do, please understand this very carefully. Now in the books they give that telling that it is going to destroy all the RBCs. The question always arises, why not the WBC? But if you actually see, they are destroying both RBC and WBC. Then what are we counting then? See, this acid, what it will do is, the glacial acetic acid, it will go and destroy or punch holes in the membrane of RBC or WBC. When the membrane is damaged, what will happen? Distilled water starts entering into the cell starts becoming larger and larger and it's going to rupture. And in RBC, do we have any nucleus over there? There is no nucleus, so we don't see at all. RBCs totally disappear. Whereas WBC, since it has a nucleus, the nucleus is remaining back. And that nucleus only we are staining using the gentian violet stain. Okay. And again in WBC now what we have to know, the normal count. Normal count and conditions when the WBC count is less than the normal, what do we call it? And when the conditions where WBC is more than the normal, what do we call it? Okay. And what are the reasons for it? Where do we see leukocytosis? Where do we see leukopenia? Reasons also you have to know in the WBC experiment. And in RBC and WBC, another thing what you have to know is the permissible difference between any two squares. In RBC experiment, the permissible difference between any two squares is 20, whereas in WBC experiment, the permissible difference between any two squares is 10. What is the meaning of this? When you take one square, let's say in one square you have 40 cells, in another square you are having 60 cells. 60 minus 40, the difference is 20. But in WBC experiment, the difference should be only 10. If it is more than 10, it shows that it is a non-uniform distribution. And what you have to do, you have to just take the preparation whatever you have made and you have to just mix it once again and charge on the Newbar's chamber. Don't be surprised that sometimes examiner may ask you the spelling of Newbar's chamber. Many students, somehow here also they find a little difficulty. So these are the very common things you have to compulsorily know. So that is about the WBC experiment. Thank you.